Hello everyone, this is Dr. Jyoti Bala. I welcome you all on my YouTube channel. In this session, we will be quickly discussing about 5 tips for designing the best primer for your PCR. So if you are planning to perform PCR, the first step is to design the primer that can specifically amplify the target sequence. Here are the 5 tips for designing the best primer for your PCR. Tip number 1, it is crucial to choose the right reason. When designing primer, it is important to choose a specific region of the target sequence which need to be amplified. This region should be unique and not present in any other genomic region, and also try to avoid regions that are repetitive or have low complexity. Depending on the needs of your experiment, sometimes you require to amplify the large fragment and the large template of DNA, while in some experiments you need to amplify the shorter template of your DNA. Moving ahead, the tip number 2 is the length matters. The length of your primer is a crucial factor. Here the optimal length of a primer is around 20-25 nucleotide. Frequently you will notice if the primer is too short, it may not bind specifically to the target sequence, whereas if it is too long, it may bind non-specifically to other regions. So make sure your primer is around 20-25 nucleotide long. The tip number 3 is check for the GC content. GC content of the primer plays a crucial role in PCR. Ideally, the GC content should be around 40-60%. If the GC content is too high, the primer may form secondary structure that can hamper the PCR reaction. And if it is too low, it may not bind efficiently to the target sequence. The tip number 4, avoid hairpins. Hairpin formation is another issue that can affect the efficiency of PCR reaction. Make sure to avoid the regions in the primer that can form hairpins or the secondary structures. To design the best primer, it is always recommended to use primer designing tool. These tools can analyze your target sequence and suggest the best primer that meet all the criteria. Some of the popular tools are Primer 3, Primer Blast and Primer Quest. So here you have it, 5 tips for designing the best primer for PCR. Remember to choose the right reasons, consider the length, check for the GC content, avoid hairpins and use primer design tools for optimum result. Hope you have liked this session. Don't forget to like and subscribe the channel and do share these information among your scientific endeavor. Thank you.